Biology students, good job on the lab today. We got a lot done, but we did not have a chance to do all of the questions, answer all the questions for the results pages of our lab. So I'm going to go over that with you quickly so that you'll be able to fill that in on your notebooks. We're going to start on page 22 on the results side after looking at the onion epidermis. Number 1, after looking at the unstained onion epidermis, describe the general shape of one cell. I would say they'd look like little bricks or little rectangles or little boxes. Number 2, describe what you see inside the cell. Well, we saw some little dark round structures, which were the nuclei, and we also saw some air bubbles. So that's what I would say we saw. Try to remember what you saw and write that in there. Number 3, what difference does the stain make in viewing the cell? Well, basically we stain cells so that we can see them better. So hopefully it made some parts of the cell easier to see by making them darker. Number 4, you should see a dark circular structure in the cells. What is this structure? Well, that is the nucleus of the cell. Number 5, here is your drawing of the onion cell, and this one is labeled, which reminds me, part of your homework that is assigned for this week is to go back to lab number 4 in your biology lab manual and read through, it's called Microscope Drawings, lab 4, page 14. And then you'll know how to label your drawings properly. This one labels the cell wall and the nucleus. If you happen to get to look at your onion cell on high power, this is a rendition of that, a drawing of that, and this is also labeled. Now I'm on page 23 of your lab manual. Number 7 on page 23 of your lab manual, the answers are not in your lab manual for this one. They are assuming that you're going to learn this from your coursework, and it's very basic. You probably already know it. 7A, what is the color of a chloroplast? Well, chloroplasts are the cells in plants that handle and produce the chlorophyll, which of course feed the plant. And we'll all remember chlorophyll is green. So this is going to be green. B, what important chemical does the chloroplast contain? Oh, look, we already said chlorophyll. And then what is the function of chlorophyll in the plant cell? Well, the chlorophyll helps to carry on photosynthesis, which of course feeds the plant. And you have a more scientific definition right there for you if you want to copy that into your book or just use your own words. Either way is fine. On the bottom of 23, they remind you that you've taken chemistry probably, or maybe you haven't, and it gives you the chemical equation for photosynthesis. And what happens is 6 water molecules plus 6 carbon dioxide will yield for you your formula for photosynthesis. You can look that up either in a biology book, it might be in there, or it might also be in a chemistry book if you don't remember how to do that. Or if you haven't learned that yet, you can just copy it down on there. Now we're on page 27 where we had the picture of the cork cell. Here's an example for you if you didn't get a chance to draw it totally in today. And here is 1 and 2. When viewing the cork specimen, do you see any internal organelles? Why or why not? Well, since the cork is dead, the organelles would have disintegrated. So there are no organelles. What you're basically seeing is just cell walls in the cork. And why does the slice of cork have to be very thin in order to see the cells? Well, cork's pretty dense. And if you can't get the light source to shine through, you're not going to be able to see the cells. So that's why it needs to be so thin. On the top of page 28 in your book, it has the picture of the starch. Remember when we scraped the potato and got the starch out of the potato? We stained it. It says, why does it need to be stained? Again, stain makes the specimen appear that was kind of a milky white or clear starchy substance. So without staining those little guys, it would have been very difficult to see. Very well done today, class. Your homework will be to read lab 4. And then to read the next 2 labs, lab 7 and lab 8 for next week. Y'all have a great week. I look forward to seeing you. Bye.